In this video, we're going to go over the genetic code. Now, the genetic code is made up out of codons, which are 3 nucleotide bases that specify a single amino acid. And since we have 4 bases at our disposal, we raise that to the power of 3, and we find out we have 64 different types of codons we can use. Now, many amino acids are degenerate. That is, they are specified by more than one codon. And when we're reading codons, we'll start from a specified point and then read them in groups of 3. And these groupings are called the reading frame. Now, the big problem with insertion and deletion of nucleotides is that they cause frame shift mutations. For example, if my codon is the big red fox ate the egg, if I delete one amino acid, this sentence no longer makes sense. Now I can do an insertion though and restore half the sentence, but still my sentence doesn't really make sense anymore. Now since we are reading in triplets, it is possible for a single gene to encode for 3 different proteins depending on where you start your reading frame from. And this is actually seen in DNA bacteriophages where their DNA, one gene can encode for multiple proteins. Now let's look at our tRNA again that we saw the structure of in last video. And what happens to make a protein is that our mRNA will bind the tRNA that contains the correct amino acid. Remember, that's bound at the 3' terminal. And the way they bind is that each tRNA contains a trinucleotide sequence that is complementary to our mRNA codon. We call this the anticodon. Then the amino acids that are carried by the tRNAs, they're going to be joined depending on what order the tRNAs bind. And that is how we are forming our protein in the ribosome. Now, how was the genetic code discovered? Well, to begin with, the way that the genetic code was discovered is that E. coli were broken up and they were centrifuged so that only DNA, mRNA, ribozymes, and enzymes and other constituent elements needed for protein synthesis were left. To show that this worked, scientists would add ATP and GTP, and the system would synthesize a small amount of protein. You could also take this system and add the enzyme DNA ACE. DNA ACE chops up DNA, so when you add this, your DNA was degraded, protein synthesis now stops because you can't make any mRNA, and mRNA was rapidly degraded. So what you can do to discover the genetic code is you can add your own purified mRNA or synthetic mRNA and determine what polypeptide was made. So for example, scientists started simple. They added an mRNA that had only U on it. They found out that this created phenylalanine, so U equals phe. Adding poly A is lyse, poly C is pro. However, poly G, that mRNA isn't actually soluble in our free system. So we couldn't make an mRNA just of poly G. So we couldn't figure out what poly G encoded for using this system. Now, separate experiments that further tried to look at this code, what they did was they took different ribonucleotides and they tested their ability to promote tRNA binding to the ribosome. Because if you have a tRNA bound, you can determine what amino acid is attached through radioactivity. So, for example, UUU. When you have UUU, you find the tRNA that has phe attached to UUU. So, with this, you could, for example, see what does UUG make. Well, when we do that, leucine is now attached to our ribonucleotide. UGU, cysteine. GUU, valine. And using this method, 50 different codons were able to be discovered. Now, there were some codons that weren't discovered through this method, and so it's either through no tRNA was bound, or we couldn't tell what radioactive signal was coming off of the amino acid. It was an ambiguous result. So to get the remaining codons, they were obtained by a chemical synthesis of polynucleotides with specified repeating sequences in a cell-free translation system. For example, you would just repeat UC, 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 and you'd get ser, lu, ser, lu. And because the reading can begin on any codon, you'd have to do various experiments determining the actual code. So for example, if our repeating structure was UAC, 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 maybe when you read the first time you'd get all tyrosines out because that's where the first reading frame is. The second time, if you start at A, you would get all threonines. If you start at the third, you would get all leucines. So you would have to design your experiment in such a way to determine, okay, what nucleotide is being read, and that corresponds to such and such amino acid. Now, after discovering all of the codons, we found some interesting results. First, like I said at the beginning, this code is highly degenerate. Only methionine and tryptophan, the two least common amino acids, only have one codon. Everything else has at least two codons. And if a codon makes the same amino acid, they are called synonyms of each other. And the code is non-random. There's a lot of order to this code. So if we look at the code, you can see that changing the third amino acid a lot of times doesn't really change the changing the third nucleotide in the codon really doesn't change the amino acid in a lot of cases. We'll get the same thing out. If you change the first codon only, you'll get a similar amino acid. So U to C, they're still nonpolar. C to A, mostly nonpolar instead of met. A to G, still nonpolar. You can see all of our charge groups and our polar groups are really all lined up here. So that's saying, well, if you just change the first amino acid, you're still going to get something that's at least somewhat polar charged. So your amino acid or your protein isn't completely destroyed. If your second position is a purine, you're mostly polar amino acids. If your second position is a pyrimidine, you're mostly hydrophobic amino acids. And there are some special codons that don't code for amino acids. These are called our stop codons. That would be UAG, UAA, and UGA. And we also have start codons, AUG, and GUG can also be designated as a start codon in some cases. Now, the genetic code is mostly universal. So this is actually a big benefit when we're doing biochemistry in the lab and we want E. coli to make human genes. They can read the mRNA, no problem, because we have the same codon, the same code. But there are some variants in the genetic code. So this is seen in mitochondria. So mammalian mitochondria can be started by AUA or UGA, and AGA and AGG are stop. The reason why mitochondria are a little different is because they actually have their own genome, they have their own proteins that can handle making proteins from the mitochondria genome, but they aren't under the same constraints as the nuclear genome, right? If you change the codons of the nuclear genome, all the proteins in the cell are now different. While in the mitochondria, you might only make like 10 specific proteins for the mitochondria, so you're not changing that much compared to the regular cell. In some protozoa, we'll also see there's a slightly altered genetic code. So it's almost universal, not quite. There are some notable exceptions to that. And with that, that ends this video. I will see you in the next video.